Welcome friends to a new session on molecular biology. In this session, we will discuss Operon concept based on Operons. Regulation of gene expression, Operon circuits in bacteria and other prokaryotes, part 1. Regulation of gene expression includes a wide range of mechanisms that are used by cells to decrease or increase the production of specific gene products, that is RNA or proteins, and is termed as gene regulation. Gene regulation is essential for viruses, prokaryotes and eukaryotes, as it increases the adaptability of an organism by allowing the cell to express protein when needed. Although as early as 1951, Barbara McClintock, who is a Nobel Prize winner in physiology or medicine, showed interaction between two genetic loci, activator represented as AC and dissociator represented as DS, in the color formation of maize seeds. The first discovery of a gene regulation system is widely considered to be the identification in 1961 of the Operon discovered by Jacques Louis Monod, who is a Nobel Prize winner in physiology or medicine in 1965, in which some enzymes involved in lactose metabolism are expressed by E. coli only in the presence of lactose and absence of glucose. A variety of mechanisms are now known which regulate gene expression at different levels, including transcription and translation. In bacteria, an mRNA is typically available for translation while it is being synthesized. This is called coupled transcription or translation. Translation in bacteria may also be directly regulated, but more commonly it is passively modulated. Let us see about induction and repression. In E. coli, synthesis of beta galactosidase, an enzyme meant for hydrolysis of lactose into glucose and galactose, has been studied in detail. If beta galactosides, example lactose, are not supplied to E. coli cells, the presence of beta galactosidase is hardly detectable, but as soon as lactose is added, production of beta galactosidase increases as much as 10,000 times. The enzyme quantity again falls down as quickly as lactose is removed. Such enzymes whose synthesis can be induced by adding the substrate are known as inducible enzymes, and the genetic systems responsible for the synthesis of such an enzyme are known as inducible systems. The situation is reversed in some other cases. For instance, when no amino acids are supplied from outside, E. coli cells can synthesize all the enzymes needed for synthesis of different amino acids. However, if a particular amino acid like histidine is added, production of histidine synthesizing enzymes will fall down. In such a system, the addition of end product of a biosynthetic pathway will check synthesis of the enzymes needed for its biosynthesis. Such enzymes whose synthesis can be checked by adding end product are known as repressible enzymes, and these genetic systems are known as repressible systems. Let us now see about inducer and co-repressor. The substrate whose addition induces synthesis of an enzyme called inducer. For example, lactose in the case of the synthesis of beta galactosidase. Similarly, the end product whose addition will check the synthesis of biosynthetic enzymes is known as co-repressor. For example, histidine in case of histidine biosynthetic enzymes. Although only a substrate acts as inducer, but rarely there are molecules that adequately resemble the natural inducers, but are not metabolized themselves by the enzyme. An excellent example is isopropyl thiogalactoside, IPTG, which like several other galactosides resembles lactose and thus has the property of induction. However, IPTG is not recognized by beta-galactosidase. Such inducers which induce enzyme synthesis without getting metabolized are called gratuitous inducer. Such inducers are very useful for conducting rDNA studies, since they induce enzyme synthesis, but remain in the cell in original form. This also indicates that there must be sites other than target enzymes which will recognize the substrate and utilize it in giving directions for enzyme synthesis. We discussed above that in the absence of lactose, no beta-galactosidase is synthesized. This would mean that in the absence of inducer, the gene or genes responsible for the synthesis of beta-galactosidase do not function. How is it brought about? It is proposed that a class of molecules called repressors is found in cells, and these repressors check the activity of genes. Ordinarily, these repressors may be active or inactive. Active repressor may be rendered inactive by addition of an inducer, while an inactive repressor can be made active by addition of a co-repressor. Let us see about the Operon model for transcriptional regulation. The basic concept for the way transcription is controlled in bacteria is called the Operon model. The Operon model was proposed by Francois Jacob and Jacques Monod in 1961 on the basis of their study on the inducible system for the synthesis of beta-galactosidase enzyme in E. coli. The Operon model has been variously modified ever since it was originally proposed by Jacob and Monod. In its original form, Operon model assumed the presence of a unit of coordinated control of protein synthesis, and this unit was called Operon as seen in this figure. An Operon consists of 1. An operator gene which control the activity of genes, 2. a number of structural genes which take part in the synthesis of proteins. This means that the structural genes will synthesize mRNA under the operational control of an operator gene. The operator in its turn is under the control of a repressor molecule synthesized by a regulated gene, which is not a part of the operon. The mechanism of regulation of protein synthesis utilizing Operon model can be illustrated using the example of two different operons, lac as well as tryptone operons in bacteria. The lac operon in E. coli. The lac operons as seen in this figure consist of 26 base pair. There are 3 structural genes in lac operon, which includes 1 LAC-Z consisting of 3063 base pair, which codes for the enzyme beta-galactosidase, which is active as a tetramer and breaks lactose into glucose and galactose to be utilized in the cell. 2 LAC-Y consisting of 800 base pairs, codes for beta-galactose permease, which is a membrane bound protein and helps in transport of metabolites, and 3 LAC-A consisting of 800 base pairs, which codes for beta-galactose transacetylase, an enzyme that transfers an acetyl group from acetyl coenzyme A to beta-galactosides. Any mutations in LAC-Z and LAC-Y genes give LAC- genotypes, which cannot utilize lactose, while LAC-Z mutants abolish enzyme activity. LAC-Y mutants cannot take up lactose from the medium. No defect is observed in LAC-A mutants. It is possible that acetylation gives an advantage for utilizing certain non-metabolizable analogues for beta-galactoside because it results in detoxification and excretion. In lac operon of E. coli, repressor is synthesized through activity of a gene LAC-I known as regulator gene. The repressor then attaches on operator gene and checks synthesis of mRNA. This is clearly depicted in this figure. If lactose is added, the repressor is rendered inactive, so that it cannot attach on operator gene and the synthesis of mRNA takes place. This is illustrated in the figure. The activity of the regulator gene is controlled by a promoter gene on its left side. It has been shown that mutations in this promoter may increase the quantity of mRNA for repressor. Lactose is not the real inducer of Operon. Since in the repressed state of Operon, E. coli cells will not synthesize beta galactoside permease, one can question how lactose enters the cell. It is believed that a minimal amount of enzyme is always present in the cell to start the metabolic pathway. It has been shown that lactose is not the real inducer of Operon, but rather it is an anti-inducer, since it binds to repressor to increase its affinity for operator. On the other hand, the bound protein in inactive repressor was allolactose. It has also been shown that while beta galactosidase breaks lactose into glucose and galactose, a side reaction changes galactose into allolactose and galactobiose. Therefore, in the regulation of Operon, lactose is taken up and a part of it through galactose is converted into allolactose, which then induces the Operon and outweighs the anti-inducing effect of lactose. Let us see about constitutive mutants. It has been found that a mutation in regulator gene LAC-I results in the absence of active repressor and consequently results in uncontrolled synthesis of enzyme. Such strains which continuously synthesize enzyme independent of need are known as constitutive strains or constitutive mutants. These constitutive strains can also be obtained by mutations in the operator gene. A mutation in operator gene does not allow repressor to act because the repressor functions by acting on operator site. Therefore, there are two types of constitutive strains: one regulator constitutive and two operator constitutive. The above two types of mutations, though have the same net result, but differ in their location as well as in their dominant recessive relationship with respect to the wild type. This relationship has been worked out by using the partial diploid cells, merozygotes. Merozygotes are those which have one complete chromosome and a part of another chromosome covering the desired region. When heterozygotes for regulator constitutive were derived, no enzyme synthesis was observed, suggesting that wild LAC-I is dominant over mutant LAC-IC. This is illustrated in this figure. On the other hand, partial diploid cells heterozygous for mutation at operator site exhibited constitutive synthesis of enzyme, indicating that normal operator gene O is recessive to mutant gene OC. This is clearly depicted in this figure. There is yet another class of regulator constitutive mutants which are dominant LAC-ID, so that in merozygotes transcription continues. This LAC-ID mutant gives repressor subunit defective at N-terminal region. This defective subunit takes part in formation of a tetramer, which is partly derived from LAC-I and partly from LAC-ID, so that the tetramer is unable to bind on the operator. Let us check about the promoter gene. While operator gene is the site of action of repressor, the actual site of start of transcription is different and is known as promoter gene, which lies to the left of operator gene. It is also believed, therefore, that perhaps RNA polymerase binds to and moves from promoter site. The repressor, when attached to operator, prevents the movement of RNA polymerase as shown in this figure. Here, the relationship between promoter gene repressor and RNA polymerase is clearly shown. The promoter gene in lac operon was inferred from mutations at this locus, which were found to affect the maximum rate of transcription. There are two proteins which are known to be involved in the regulation of lactose operon. One, lac repressor, and 2, catabolite gene activator, that is CGA protein, also known as CAP, that is cyclic AMP protein, or CRP, that is CAMP receptor protein. While repressor binds to operator site, CGA protein binds to CGA site. Moreover, repressor exercises a control by checking RNA polymerase to travel through operator, but CGA protein exercises a positive control. Only when CAMP molecule activates CGA protein, the latter will allow RNA polymerase to bind. This is clearly shown in this figure. A protein such as CAP, which interacts with the DNA and RNA polymerase to assist in transcription initiation, is called apoinducer. It has been shown that CAMP is essential for activation of CGA protein, since when CAMP was removed or was absent due to mutation, transcription was reduced. In mutants lacking CAMP, transcription was restored by adding CAMP exogenously. It has also been shown that this CAMP CGA protein system is influenced by the level of glucose concentration. It is therefore sometimes described as glucose sensitive system, which is common to several glucose sensitive operons in E. coli. Example, operons for degradation of lactose, galactose, arabinose, maltose, etc. In all cases, glucose is one of the catabolic products of biosynthetic pathway, and it accumulates, it becomes very injurious. Therefore, whenever the concentration of glucose increases beyond a certain level, it leads to reduction of transcription. This is achieved by reducing the level of CAMP by glucose, either through checking the synthesis of adenocyclase, an enzyme needed for the synthesis of CAMP, or by removing CAMP by making a complex with it. Therefore, transcription in lac operon requires 1, CAMP activated CGA protein, that is positive control, and 2, lac repressor inactivated by inducer, that is negative control. Operator and promoter are cis dominant. It should be realized that in the lac operon, operator can control only lac genes that are adjacent to it. If a second lac operon is introduced as done in merozygotes, none of the operators is influenced by the other operator. In a partial diploid, one operon has normal operator O and other has a mutant operator OC. The operon with OC will be expressed, but with O will not be expressed. This could be tested by using the partial diploids, which not only differ in operator, but also differ in structural genes. For instance, in a partial diploid with genotypes OC Z plus Y minus and O Z minus Y plus, only beta galactosidase will be synthesized, but not the beta galactoside permease, although Y plus is present along with OC, but on 2 different chromosomes. This phenomenon of lack of dominance extended to other chromosome is called cis dominance. This also suggests that operator does not act through a gene product like regulator gene, which acts through repressor, which can diffuse and influence the other allele also. This phenomenon will be expressed by all those sites which are physically contiguous with the sequences of controls and function by being recognized rather than by giving a gene product. It is for this reason that promoter and terminator are also cis dominant or cis acting, while regulator is trans acting. Association and dissociation of Lac repressor with the operator. The binding of purified Lac repressor protein to DNA was first characterized by a filter binding assay. This is based on the principle that nitrocellulose filter retains protein or DNA complexed with the protein, but not pure dsDNA. Thus, the DNA sequences of operator used by repressor for binding could be identified by their retention on the filter. It was shown that repressor could not bind on DNA obtained from a OC mutant. The location of binding in Lac Operon extended from -5 to +21 within the transcription unit, thus overlapping the right end of promoter. This overlapping does not occur in all operons. Although earlier it was believed that binding of repressor does not allow binding of RNA polymerase, now we know that both can bind simultaneously, but RNA polymerase cannot initiate transcription unless repressor is released by an inducer. Repressor is a polypeptide of 360 amino acids and is divided into two regions. One, 1 to 59 amino acid residues on the amino terminal and makes a long headpiece which can be cleaved at position 51, and the fragment 1 to 51 is called short headpiece. 2, 60 to 360 amino acid residues on the C terminal end make trypsin resistant core, that is trypsin cleaves the repressor at amino acid number 59. The long and short headpieces both can bind to operator independently of the core, and therefore, it is assumed that their structure does not change in association with the core. When lactose inducer is added, repressor will dissociate from the operator. This is explained on the basis of several models. 1. Repressor is normally in equilibrium between associated and dissociated state. When the inducer binds to dissociated repressor, it is rendered incapable of association. 2. Inducer may bind to repressor associated with operator and changes the conformation of repressor, which allows it to get off the operator. 3. Inducer changes the relative conformation of head and core regions of repressor, so that headpiece cannot hold the DNA tightly. 4. The repressor in tetrameric form may be in excess and remains associated at other random sites on DNA, but is not found free within the cell. Due to induction, when the repressor is dissociated from Lac operator, it is stored at other random sites and used again when needed. The effect of induction therefore is to change the distribution of repressor on DNA and not to release free repressor. DNA bending during binding of CAP protein. During early 1990s, DNA protein complexes have been analyzed to find if DNA geometry is altered when a regulatory protein binds to it. It has been shown that DNA bends considerably when the regulatory protein CAP binds to promoter site. Similar bending has been observed during binding of zinc finger proteins or leucine zipper proteins or TF2D. This bending of DNA is very important in activation of transcription. In case of CAP, bending due to CAP can be about 90 degree, but the protein-protein interaction between CAP and RNA polymerase leads to formation of a nucleoprotein complex showing a DNA bending of up to 180 degree. The CAP protein consists of 209 amino acid residues with the 2 domains. The carboxy terminal domain of the protein, which is involved in DNA binding, is shown in this figure. CAMP is bound only to its larger amino terminal domain. Each CAP molecule has 3 alpha helices, alpha D, alpha E, alpha F, and the distance between alpha F helices in a dimer is 34 Armstrong units, making it possible for them to fit into the grooves of B DNA, which may take the form of Z DNA during DNA protein interaction. CAP has been compared with other proteins like lambda repressor and lambda crow-protein, showing similarities in structural details and amino acid sequences in DNA binding domains. Comparisons of these 3 proteins, CAP, lambda rep and lambda crow, revealed a conserved recognition motif consisting of alpha helix, a turn, and a second alpha helix. These are therefore described as helix turn helix or HTH proteins. Let us summarize the following discussions on Operon. A variety of mechanisms are known to regulate gene expression at different levels, including transcription, processing of mRNA and translation. The basic concept for the way transcription is controlled in bacteria is called the Operon model, proposed by Francois Jacob and Jacques Monod in 1961 on the basis of the study on the inducible system for the synthesis of beta galactosidase enzyme in E. coli. The unit of Operon model is called Operon, consists of an operator gene and a number of structural genes. The Operon consists of 26 base pair and is a part of the first structural gene LAC-Z. There are 3 structural genes in Operon, which includes gene LAC-Z, which codes for the enzyme beta galactosidase, gene LAC-Y, which codes for beta galactose permease, and gene LAC-A, which codes for beta galactose transacetylase. Now, I will give you certain assignments based on the discussions what we had. 1. Compare and contrast repressible and inducible operons. 2. The Operon is an inducible operon. Why? 3. Describe the process of induction and repression in the Operon. 4. Lactose is not the real inducer of Operon. Explain. 5. The Operon considered to be an example of transcriptional control. Why? I will also give you certain references for your further reading. First one is a book with the title Biochemistry, 5th edition, authored by Berg, Timsko and Stryer in the year 2008 and published by Freeman and Company, New York, USA. A second book is with the title Molecular Biology of the Cell, 5th edition, published by Alberts, Johnson, Lewis, Raff, Roberts and Walter in the year 2007, published by Garland Science, New York and London. A third book is with the title Gene 9, authored by Levin in the year 2007, published by Johns and Bartlett Publishers, Sudbury, Massachusetts. A fourth book is with the title Biochemistry, authored by Davidson and Sitman in the year 1997, published by Waverly, New Delhi. A fifth book is with the title Principles of Genetics, authored by Sinnett, Dunn and Dobzhansky in the year 1958, published by McGraw-Hill, New York. I will also give you certain websites for your further reference. The first one is www.bioencyclopedia.org. The second one is www.oxfordgenetics.com. We have come to the end of this section. We will meet in another session with a new topic. Until then, bye.